Good afternoon, everyone. Welcome to today's Public Health Ontario rounds presentation on launching the novel torpedo surveillance method for avian influenza viruses in wetlands. My name is Dr. Richard Mather. I'm a public health physician at Public Health Ontario, and I have the pleasure of moderating today's session. Before we begin, I will mention a few housekeeping items. First, I'd like to declare that as the moderator of this session, I have no conflicts of interest to declare. If you require CME credits, please email capacitybuilding@oahpp.ca. It is now my pleasure to present our speakers for today's event, Dr. Samira Mubareka, Dr. Claire Jardin, and Juliette Blais-Savoie. Dr. Samira Mubareka completed her medical degree at Dalhousie University and internal medicine training at McGill University in Canada. She specialized in infectious diseases and medical microbiology at the University of Manitoba and went on to do a research fellowship at the Mount Sinai School of Medicine in New York City. Samira is currently a virologist, medical microbiologist, and infectious disease physician at Sunnybrook Health Sciences Centre in Toronto, Ontario, and in the Department of Laboratory Medicine and Pathobiology at the University of Toronto. She is currently focused on understanding the biology and transmission of SARS-CoV-2 variants of concern and on coronavirus and influenza virus zoonotic spillover. Dr. Claire Jardin is an associate professor at the Department of Pathobiology and the regional director of the Canadian Wildlife Health Cooperative for Ontario and at the University of Guelph. Dr. Jardin completed her Master of Sciences degree in ecology at the University of British Columbia, her doctorate of veterinary medicine at the Western College of Veterinary Medicine, and professional doctorate degree at the University of Saskatchewan prior to her appointment at the University of Guelph. Dr. Jardin's research focuses on wildlife health and disease with a particular focus on investigating the ecology of multi-host pathogens in wildlife populations. Juliette Blais-Savoie is a second-year graduate student at the University of Toronto's Department of Laboratory Medicine and Pathobiology and Sunnybrook Research Institute, supervised by Dr. Samira Mubareka and Dr. Nicole Midio. Her research focuses on using viral genomics to study influenza A viruses with a primary focus on avian influenza viruses. In addition to avian influenza surveillance, she is also conducting studies on the evolution of human, seasonal, and avian influenza viruses through different propagation methods used for vaccine manufacturing. I will now hand things over to Juliette to begin the presentation.
Thank you, Dr. Mather, and welcome everyone to our presentation on launching the novel torpedo sampling method for avian influenza virus detection in wetlands. Before we start, just a disclaimer that this presentation was created by myself and the other presenters and is in no way reflective of the opinions of Public Health Ontario. And we have no conflicts of interest to disclose. So for today's learning objectives, hopefully at the end of the session, everyone will be able to describe both basic avian influenza virology and the viral ecology. You'll be able to interpret the results of our study on the efficacy of this novel surveillance method and the impact that it can potentially have for surveillance efforts, and be able to identify risk factors for avian influenza in humans. And with that, I'll pass it over to Dr. Samira Mubareka for a little bit of an introduction on the virus.
Thank you very much, Juliette, and I apologize to everybody in advance. We're kind of in the middle of sampling in the field, so I'm sitting in my vehicle and hopefully the connection will hold, but if I do drop off, I apologize. So my role today is really just to give you a fairly quick primer on avian influenza viruses with a little bit of emphasis on highly pathogenic avian influenza virus. And I think probably one of the most important things to note is that migratory waterfowl as well as shorebirds are really the main reservoir for influenza viruses. Apart from H17 and H18, and that's how many hemagglutinin subtypes we're up to now, those two are found exclusively in bats. All the other ones have been detected in migratory waterfowl and other birds. But as you can see from this slide, and maybe paying attention more to the left-hand side, specifically looking at influenza A viruses, there is quite a plethora of different mammalian species that these viruses can infect, and obviously that includes humans as well. As you all know very, very well, all too well, that seasonal influenza viruses have continued to persist. Now, the reason we worry about these viruses is not only for their capacity to burden the healthcare system, cause mortalities, in livestock, cause mortalities in wildlife, but also because of their pandemic potential. And pandemics tend to emerge when there's an antigenic shift. So these viruses are brilliant in the sense that their genome is segmented. So if you have two different viruses that infect the same cell, you can have segments from those two different viruses that get packaged together. So even though packaging is selective in terms of, or in the sense that there's one of each segment in every virion, they could be from different viruses, usually two different ones, if not just a single one. And that's really when we see antigenic shifts emerge. So when the HA or the NA changes, and a pandemic, if it is something that spills over into humans, is pathogenic in humans, and can be transmitted among humans, then pandemics tend to emerge. So this is just an example here on the right, where an avian H3, we don't know what the NA was, reassorted with an H2N2 that had been circulating on a seasonal basis back in the late 1960s, that caused an antigenic shift. So that was the first time that humans saw H3 viruses. That caused a pandemic, but then that virus persisted as a seasonal virus and just gently drifted. So that means that there were some single changes in the HA, and that's why we update the vaccine every year, but really no major shifts. Now, the one virulence factor I want to draw your attention to, because it's really important in terms of distinguishing low pathogenicity avian influenza viruses and high pathogenicity avian influenza viruses, is the hemagglutinin. So the hemagglutinin is really important on so many different levels. First of all, it harbors at least 5 antigenic sites that are quite critical in the globular head. It also harbors the receptor binding site, as you can see on the slide on the right. So that's important because alpha-2,3 linked sialic acids predominantly or preferentially bind avian influenza viruses. Alpha-2,6 linked sialic acids preferentially bind mammalian influenza viruses. So you can already see some species specificity based on this biology. But what really makes the difference between low and high pathogenicity avian influenza virus is a change in the stalk region. So that's lower down. And there's a cleavage site there. So cleavage allows the virus to get into the cell. And usually, if it's not polybasic, it's really restricted to serine proteases. If it is polybasic, as we see in highly pathogenic avian influenza viruses, much more ubiquitous enzymes like furin can cleave it. So that enables the virus to replicate in a much broader range of tissues. So transitioning from maybe the more molecular biology to a little bit more genomic epidemiology, because things really have changed in terms of the H5 viruses that are circulating right now. We know that H5N1 and H5NX, so other N types, have been circulating for decades now, since the 1990s. But what's happened recently, especially since 2005, is a change in the hemagglutinin. So we're seeing reassortment with the other NA types. So for example, at one point it was H5N8. This has now, if you're just looking at the bottom figure, now been supplanted by H5N1 in the lighter green. But it's also the subclade that really seems to have taken over, the 2344B subclade, that is reassorting substantially with internal genes. So even in Canada, even though we've had predominantly of the H5s, or mostly N1s, a lot of those internal genes are reassorting all the time. The other thing that has changed is that the geography has changed substantially. So in the past, earlier on, we mainly saw H5 viruses circulating in Asia. If you look just to the right of this phylogenetic tree, you can see a little bit more green, where we're seeing more circulation in Europe and then pink in North America. The other thing that shifted is it's gone from mainly domestic birds, so poultry, where you can control viral activity by stamping out strategies, to wild birds. So you're seeing more and more blue bands over time and more recently. And what's happened after the virus landed in the Americas, actually here in Canada and Newfoundland, it seeded the rest of the Americas from there. And it's actually, unfortunately, gotten to the point where it has reached Antarctica. So it has spread globally from multiple sources, pretty much everywhere around the world except for Oceania. And unfortunately, one of the other red flags about this virus is that it's spilling over into more and more mammals, and it includes marine mammals, as you can see in the pictures here. And if we take a little bit of a closer look at the disease it's causing, we're seeing necrotizing encephalitis, which is something that is probably responsible for the high fatalities in some of these mammals. And of course, it's something that flags to us as human health people because of the concern for that possibility as well. Having said that, what we're actually seeing, and this is obviously in a very limited number of individuals, is H5N1 infection in dairy farm workers that presents more as hemorrhagic conjunctivitis. So that could have to do with the exposure, it could have to do with the biology, but this transmission of H5N1 viruses among dairy cattle is very, very concerning because it's really the first representation of unequivocal, clear mammal-to-mammal transmission. It may or may not have been happening among the marine mammals, but now there's no doubt whatsoever. So that really represents a difference or a change in the viral ecology. And part of the reason that these patients are presenting with hemorrhagic conjunctivitis, you can see on the right here, there are human corneal epithelial cells that, this is older data, so this is not, these findings are not new, but they support an older, so this is the Vietnam H5, they support replication of H5 viruses. And that's because we not only have alpha-2 3-linked sialic acid in the lower respiratory tract. So that's in pink. And remember, those are the ones that bind avian influenza viruses preferentially. We also have them on our eyes. In the upper respiratory tract, denoted more so in green, are the alpha-2 6-linked sialic acids. So those are the receptors that are preferentially bound by mammalian influenza viruses. And this is just my last slide before I hand it over. Just to note that often we think about the mortality with these cases as being quite high. You hear numbers like 40%, 50%, and this is just a reminder that these are based on fairly small studies that are from some time ago. So these are not with the currently circulating subclade. So we really don't know what the mortality would be for the H5N1s that are 2344B subclade. But regardless, we certainly can't be complacent because looking at these numbers, you're seeing these patients are relatively young, and despite that, having high rates of both respiratory failure and very high rates of mortality. So with that, I will hand it over to Dr. Jardine to tell you about surveillance.
Awesome. Thank you so much, Samira. That's great. So I'm just going to start just with a map because I love the maps, and I just wanted to show you a little bit about sort of set up this, why we do the surveillance the way we do. So this particular, as Samira mentioned, this particular H5N1 avian influenza virus, clade 2344B, emerged in Asia, spread to Europe, and then to North America, associated with the movement of wild birds. Now, we don't typically think about wild birds migrating across the Atlantic, but there is a meeting point for these birds in Iceland. So they'll go there and you'll have North American birds and European birds together in those areas. And then it seems likely that the virus spread from Europe, from these European birds to North American birds at this time. And then the North American birds came back to North America. And this map, you can see along the Atlantic coast of North America, those little red dots represent the first incursions into North America. And this is as of January 2022. And then, you know, it's interesting to think about how these cases on the Atlantic coast then led to the spread of avian influenza across North America. And it's, you know, we know that it is through the movement of wild birds. And generally we think about, you know, waterfowl moving along these flyways. So we think about north-south movements. But you can see from this figure here, there is a lot of overlap between the flyways, and birds are able to mix. And that's what allowed the virus to spread across the continent in a relatively short period of time. And as Samira was mentioning, you know, this is the current global distribution of the virus, and it now includes incursions into Antarctica. And as these, you know, as this, you know, this virus spreads, it has opportunities to mutate. There's opportunities for these reassortments and this antigenic shift. And there's a mixing of viruses like these low-path avian influenza viruses that are in our North American wild birds are able to mix with this high-path strain as well. So all this spread and mixing allows for a lot of changes to the virus. So this, I just wanted to show you these. This is the avian influenza dashboard. You can go to it. It's on the CFIA has a link to it. You can see the detections here. The stars are domestic animal birds, and then the circles are wild samples. And you can see there's lots of cases in Southern Ontario. And I thought it would be interesting to sort of describe how we get these dots in terms of the wildlife surveillance, just so you're aware of that. It helps to interpret and understand some of the biases that are associated with this surveillance that we do in wildlife. So we've actually Canada has been conducting surveillance for avian influenza since 2005 through this interagency wild bird influenza survey that we've been doing. It's a collaborative effort between federal, provincial, and territorial government agencies, non-governmental organizations, and academic institutions. And the program was put in place with two main objectives. Firstly, to expand our understanding of which avian influenza viruses are circulating wild bird populations, and then also enhance our ability to detect incursions of highly pathogenic avian influenza strains. And we conduct this surveillance through using two main sampling strategies. We test live birds to look and better understand which influenza viruses are circulating, and we test dead wildlife to try to do a better job to detect these incursions of highly pathogenic avian influenza. So in Ontario, the Canadian Wildlife Health Cooperative is the primary point of contact for people who find dead animals in the province, dead wild animals in the province. And we rely on opportunistic submissions of dead wild animals. Most of our submissions, it seems weird, how are we getting these dead animals in? But government agencies, wildlife rehabilitators, the public as well will call us and submit carcasses. And these animals that come in are then tested for avian influenza. And this photo here is a dead Canada goose that was found and tested positive for avian influenza, just to give you an example. The other component, which is geared towards understanding which avian influenza viruses are circulating in wild bird populations, we use live bird surveillance. I just wanted to show you a picture of that. So this is airboat sampling. Healthy birds are captured, they're swabbed. So you can see this person is swabbing a duck, getting an oral pharyngeal swab. And that's airboat sampling that's done with the Ministry of Natural Resources here in Ontario. So then all these swabs are submitted to our provincial animal health diagnostic lab, which for us in Ontario is the Animal Health Lab at the University of Guelph. And then any matrix H7 or matrix H5 and H7 positive or inconclusive samples are then sent to the National Center for Foreign Animal Diseases for confirmation and identification. And that information is then shared by the CFIA with the World Organization for Animal Health as required. So I thought I'd just show you, a little bit of the data and outcomes of the surveillance that we do at the CWHC. So that first map I showed you of surveillance in Ontario just showed the positives. This map here shows all the animals that have been tested in 2023. The yellow dots are the negatives and the reds are positive, just to give you an idea of the sort of distribution of the surveillance. And you can see we have pretty good coverage of some areas of Ontario, but there certainly are gaps in our surveillance, geographic gaps in our surveillance. And I also wanted just to quickly show you kind of the temporal surveillance that we've been doing. So it's the total number of cases is the height of the bars. The orange are positive and the blue are the ones that have tested negative for avian influenza. In Ontario, the first case associated with this current outbreak was detected in a red-tailed hawk in February in 2022. And you can see there's definitely seasonal changes in the occurrence of the virus. So if you look at those orange bars, and these changes are likely impacted by a variety of viral host and environmental factors. But it's also important to notice that, there is, changes in the surveillance effort over time. So it reflects a little bit of the opportunistic nature of our surveillance program. All right, and so I just wanted to put this graph. So we are, we're looking in wild birds, we're looking in wild mammals as part of our surveillance. And I think, obviously these approaches are really valuable. They provide a lot of important information, but they have limitations in terms of our ability to acquire samples from areas that are of particular concern, which may be the case for some of this farm situation up in the in the top of the picture there. So we can't control the location of dead wildlife, and sampling of live wildlife. It's expensive and really labor and time intensive, and we do small amounts of it, but all these factors kind of limit the geographic scope of our testing. So one solution to these shortcomings is to do more directed detection, or to do directed detection directly from the environment, like water. And that kind of brings us to our research project. And so now I'm going to turn it over to Juliette to tell you about that. Thank you.
Thank you, Dr. Jardine. And so as Dr. Jardine just highlighted very well, although there are a lot of really wonderful efforts being conducted by the CWHC and other groups for direct animal sampling, there are a lot of limitations with this method. And an environmental method that some of you may be familiar with is sediment sampling, where for wildlife, one would collect sediment from wetlands where wild waterfowl are nesting and test those samples. Sediment samples are very, very complex, and it can be a real challenge to get good data out of them, both by PCR or by sequencing. And that brought us to our novel device, which we term the Torpedo. This device was designed by Dr. Edward McBean's group at the University of Guelph School of Engineering. It is 3D printed, very small, compact, portable, and when it is filled with sorbent materials, as you can see here in the picture on the left, it can be used to detect virus that is present in water. And it was used with pretty substantial success during the COVID-19 pandemic for wastewater surveillance for SARS-CoV-2. For our study, we wanted to test the efficacy of this device in detecting avian influenza virus in different wetlands throughout Ontario. So to do this, we tagged onto existing efforts from the Ministry of Natural Resources and Forestry for banding wild waterfowl. So as field research teams were out netting wild waterfowl at different wetlands in the province, we had the Torpedo device dragged behind the boat that they were using to passively sample the wetlands where these wild waterfowl were nesting. And as the tagging efforts were done, we also had the field team collect oral and cloacal swabs from these animals, which gives us a really fascinating data set of the waters at different wetlands and the animals that are present at those wetlands, which should theoretically be shedding the avian influenza virus into these water systems. We then did downstream analysis at Sunnybrook Research Institute, beginning with RNA extraction, followed by quantitative PCR with H5, matrix gene, which is a universal influenza A target, and also an internal extraction control target. Afterwards, all positive samples underwent whole viral genome sequencing on the Illumina mini-seq platform. And here are our results. So as you can see, we sampled 6 different wetlands throughout the province of Ontario. And of those 6 wetlands, we were able to detect avian influenza virus at 5. Of these 5 sites, we only got torpedo detection of avian influenza virus at 4, but that is still a fairly decent correlation with presence or absence data. Additionally, we found 1 H5 positive and 3 H5 inconclusive samples at the Matchdash Bay site up here. And our results were separated into positive, indeterminate, and negative. Indeterminate just being samples that had some signal for influenza A RNA, but did not meet our threshold for positivity. Once we had these PCR data, we wanted to investigate which of the sorbent materials that we had packed into the Torpedo performed best. And as you can see, out of the 3 materials, gauze pads, Swiffer pads, and polycarbonate filters, the first 2, gauze pads and Swiffer pads, performed basically the same, each with 8 individual samples testing positive, while we got no viral RNA detection from any of our polycarbonate filters. After PCR, we went on to attempt whole genome sequencing with our positive results, and we were able to obtain a total of 36 whole viral genome sequences, some of which from the waterfowl swabs and some of which from the torpedo samples, which was very exciting that we were able to get high enough sample qualities out of this device that we were able to obtain whole viral genome sequences. This can be a real challenge with environmental sampling. And looking at over the data, we detected a total of 7 different unique low pathogenic avian influenza viruses, not including the H5 positive samples that we discussed previously. And notably, at this site, Boat Lake, we were able to detect the same subtype, H6N2, both in our waterfowl samples and in our torpedo environmental samples. And this is really promising because it shows us that the torpedo device is capable of detecting the same subtype that is actively infecting animals at a particular site that they should be shedding in real time. Once we had these sequences and we had done the base subtype analysis, we sent them to the National Center for Foreign Animal Disease, to Dr. Anthony Signor, so that we could get an idea of how our samples compared to H5 HPAI sequences that had been collected all across Canada, which the NCFAD has access to. And Dr. Anthony Signor very kindly helped us create these phylogenetic trees for several internal genes. So the first one I'm showing you here is the PB1 gene. And as you can see here, the blue and green colored branches are our samples from our study, low pathogenic avian influenza virus. And all of these pinkish red samples are H5. So we are seeing relationships between the internal genes present in our low pathogenic avian influenza virus sequences and those present in H5 sequences as well. For this gene, PB1, we tend to see still some level of differentiation. The LPAI PB1 genes tend to be clustering together and the H5 ones tend to be clustering separately with a slightly more ancestral common ancestor. However, when we move forward to PB2, it appears a lot more intermixed. We are seeing our low path sequences cluster with high path sequences within a variety of different subgroups. So you can follow these subgroups with these black lines. And as you can see, we have a mixture of low path and high path in subgroup B, in subgroup J, subgroup H, and subgroup 1. So this appears to be showing a lot more intermixing with the PB2 gene. And overall, we saw reassortment in 2 to 4 internal genes for all of our low pathogenic avian influenza virus sequences. So this is showing relatively high rates of reassortment between the low path sequences that we were able to detect in Ontario and the high path sequences that are being detected across the country. So in conclusion, I just want to go over the major findings of the study. First of which is that the torpedo environmental sampling device is capable of detecting avian influenza virus in a field wetland setting. And also the fact that we were able to obtain sequenceable viral RNA out of this device. The sequences revealed to us that we are seeing a lot of reassortment between the low pathogenic avian influenza viruses present in Ontario and H5 sequences that are being detected across Canada. And this gives us a really good idea of the ecology and reassortment and evolution that is happening in the context of this current outbreak, which Dr. Mubareka and Dr. Jardine outlined at the beginning of this presentation. And so finally, we hope that this device will be very applicable to helping fill some of the gaps that we discussed in avian influenza virus surveillance, both for H5, but also for these low pathogenic avian influenza viruses that, as we've shown, seem to be contributing to the evolution of this outbreak. So this device could be used passively by holding it or tying it stably in a flowing body of water. And this would allow us to have really targeted use, particularly around poultry or dairy farms, which have been having massive outbreaks in the United States. And we're also seeing poultry farm outbreaks in Canada. And this helps us get an early idea of what's going on around these agricultural sites where we don't see a lot of wild animal sampling. Additionally, the device is very lightweight, and so it has the potential to be used with drone or other type of remote technology to sample sites that are very inaccessible and very challenging for a whole research team with a boat to get out to. And so we hope that this study will really help bridge the gap and provide a tool that will help further surveillance effort for this virus of really significant public health concern. And finally, on behalf of all of the speakers, I'd just like to thank all of our collaborators on this project. It was a hugely collaborative effort and wouldn't have been possible without anyone listed here. We'd like to thank our funding sources and Public Health Ontario capacity building team for inviting us to speak today. And with that, I will hand it back over to Dr. Richard Mather for the Q&A.
Thank you to our presenters. We will now move on to the Q&A segment of this event to address some of the questions from our audience. Start with the first question. Is there a higher incidence of avian influenza in migratory birds compared to domestic birds, I think?
I guess, yeah, I can start and then I'll let Juliet and Samira add on. So I think that in wild birds, the story has kind of changed. So we see that like wild waterfowl are typically considered to be the reservoir of these, as Dr. Mubaraka said, like of low-path avian influenza. With the emergence of this highly pathogenic strain of avian influenza, we see now mortality in wild birds, and it's affecting raptors and corvids in addition to waterfowl. So it's sort of expanded the host range, I guess, of the virus, and we're seeing actual like impacts on like individual birds dying of this virus, which we didn't typically see before the emergence of this strain or clade of the virus. So I think like I think we're going to see that it's maintained in wild birds now, and there's potential for it to be transmitted from wild birds still to domestic poultry. But instead of wild birds being involved in transmission of low pathogenic strains into poultry, where they then change into highly pathogenic strains, we're going to see maintenance of this particular highly pathogenic strain in wild birds, which adds a whole new dimension. And I think I'm sort of off topic a little bit, but I guess it's a little bit complicated in terms of determining what the incidence is in wild birds. But certainly, like we've seen a lot, like out of birds that are submitted to us, we see a lot of cases in these corvids, raptors, and then in waterfowl. But waterfowl are also able to survive infection, which is why the live bird sampling is also really valuable. So these are animals that are, apparently healthy, but we detect the virus in them. So it's you can, I don't know if Samira or Juliet have anything to add, but I'll stop there.
I think the only thing I would add is just underscoring the importance of investing in live bird sampling, because as you pointed out, Claire, those are the animals that probably harbor most of these viruses, but they don't show any clinical signs and symptoms. And there are investigators like Kathy McGraw have started looking into differences in avian innate immune response, because, many of us on this call are human health people, and we don't think about that, but there are actually significant differences that then impact viral ecology. And I think that's something that's been underinvested in over recent decades, but has significant bearing on the health of both humans and other animals.
Yeah, and I've just maybe add one more part. Like, I think, we see in the dead bird surveillance, we see like, it's a lot of raptors being affected, a lot of corvids, but if there weren't waterfowl maintaining it, we wouldn't see that. I think it's, if you think about the reservoir of infection, it's really waterfowl that are primarily responsible. And then these other species, including mammals, are being infected as sort of a consequence of that. But this is all changing, right? So now, it can all change because the virus can change so rapidly. So that's how we sort of see it now, but it's not that it won't change in the future.
Just as a final point for those that might not be fully familiar with bird ecology or this virus specifically. The wild waterfowl are migratory, but wild gulls are typically associated with intercontinental spread. So when we're looking at these major jumps between, say, Europe and North America, it's typically these these marine gull species that are associated with that.
All right, thanks for all those responses. Moving on to the next question. There are some advancements towards development of rapid tests. Could you see this surveillance tool being coupled with rapid tests in the future to inform people who may be hunting wild birds?
I think it's a great idea. I think it would be really interesting. We just need to make sure the rapid tests are good enough to be used in this way. And at the moment, I don't think we have a tool in Canada that can be used in this way. There is some surveillance going on in birds that are hunted through Environment and Climate Change Canada. They're doing some surveillance using hunter-killed samples, and hunter harvested samples are sort of considered to be like live bird sampling because those were healthy at the time of harvest. So that's how we sort of include those in kind of that live or healthy bird sampling. So yeah, there is work being done in that, and I'm really interested in these rapid test kits as well and how we could apply them to wildlife. But I think there may still need some work. Some work I think still needs to be done. Maybe there's a suitable test for, a chicken or a turkey, but if you look at the suite of wild species that we want to be able to test it in, each of those, like each different species needs to be, there'd be a lot of validation, a lot of work that would need to be done before we could really trust those tests and use them in a capacity outside of maybe a research project. But I'll turn over to Sam and Juliet now.
Yeah, so just specifically for this study, the data that we have are PCR data, so they're based off of viral RNA. I don't believe there are any, and Sam, please correct me if I'm wrong, any rapid tests on the market that are RNA based currently. They're mostly antigen based, and we didn't do any antigen tests with our environmental samples. So we can't really speak to, with what's currently marketable, how this device would be able to perform. But there are people trying to develop more rapid tests than PCR that are RNA based. Some people in our lab actually are working on that. And so it does have the potential to, in the future, as technologies continue to advance, be used in a more rapid setting rather than having to be shipped to a site like Sunnybrook, where we have to do all of our extraction and PCRs in a really CL2 lab-based setting.
Thanks, really interesting. Next question, has there been any comparison of the torpedo method with a more traditional recreational water sampling method?
Yeah, so no, is the short answer. Basically, all of our comparison that we've done has been with the most traditional and most highest quality sample producing method of direct animal sampling. It's certainly something that we've discussed and consider in the future, is comparing this device to some other sampling methods. And so as we continue to conduct studies of this device and investigate potential ways of use, that definitely is a direction that we're considering going.
All right, thanks. Next question. Any potential opportunities to use this method in other diseases that may cross animal-human boundaries?
I can take a first stab at that question. I think avian influenza is a really good example because the virus is actually transmitted by the fecal-oral route between birds. So essentially, they're excreting this virus in fairly high amounts into the water. So sampling the water does seem to be fairly low-hanging fruit. I do know that others, and this is not something we're involved in, so I can't really speak to it in detail, but people have thought about doing it for AMR, particularly sort of at that livestock-human interface. But apart from that, and I'll let maybe Claire, Juliet, if they know of other examples, I'm not aware of any use of wetland sampling anyway for other viruses or pathogens.
No, yeah, and I'm sure everyone's already aware of the torpedo sampling in this sewer system. That is the same sort of technology that's been used for SARS-CoV-2 testing. That's the other, not obviously in the wetlands, but in the sewers with that device, though. I just wanted to mention that. That's how I think that device was really, that's what it was developed for. And then Juliet sort of applying it in this wetland kind of situation.
All right, thank you. Question about the strain information. Did you observe any co-infection with different strains? You mentioned some birds had H5N1 and others different strains.
So no, all of our sequences, we did some analysis to look beyond just the consensus sequence, and we found no evidence of co-infection. That might be reflective of the sample quality, but insofar as the data that we have, we found no evidence of that.
All right, thank you. Next question. How do you see this information informing preparedness and response to a potential avian influenza outbreak in humans?
I think for that, it would have to be implemented fairly broadly for it to be effective in terms of predicting potential spillover, but you still always need to have to come back to some form of risk assessment. A lot of times it's experimental to understand, especially if there's a novel reassortant that gets picked up. But regardless, each individual reassortant has to be risk assessed. So in and of itself, it could maybe be a trigger to go on and do a risk assessment, but I don't know how predictive it would be on its own, if that makes sense. And it also depends on what the interface looks like, right? So it's not only about detecting it, but how proximal, what the exposure would be, obviously. So a much more extensive risk assessment beyond experimental, but also epidemiological, if that helps. I don't know, Claire, if you have anything to add to that as well.
Great. I was just saying, I think that was a great answer. I don't have anything to add. Thank you.
Thank you. A follow-up question. Are there any plans to do serial testing at certain sites to look for potential evolution of viruses over time? And also, any thoughts on how many samples would be needed across Ontario to be able to give a signal for Ontario, same way that we had done for wastewater signaling for SARS-CoV-2?
I can start. So far as the serial testing goes, because we are sort of piggybacking off of the existing banding efforts, our ability to revisit sites is really dependent on whether the MNRF is planning on revisiting sites for banding efforts, and that's entirely possible that they will be doing that. But unfortunately, we can't really build that into our study design. And then I think Dr. Jardine would probably be best to answer the overall signaling part of that question.
Yeah, and I think just this was a sort of smaller pilot project that we were just sort of seeing if the method would work. And I think it would need, obviously some more research and I think there may be groups that are starting to look into that now, using this tool in the future. So I think I'm not sure that it's ready to go like now, but it's like sort of maybe we're kind of moving it along, I guess. Yeah.
Thanks. One more related question about sampling. Is there a certain time of year when it would be best to do sampling, perhaps to align with the migratory bird patterns or flyways? Yeah,
that would make a lot of sense to me. Like right now in our dead bird surveillance, we're doing, surveillance is ongoing and we don't get a lot of detections over the course of the summer. This virus tends to like cooler times of year and it obviously coinciding with migration, right? So nobody wants this to happen, but we expect probably that we'll see cases again this fall. Last year we had cases over the winter in geese over the winter, and then a little peak in the spring, and then goes down. So I think we just want to make sure if we have other types of surveillance, we want to make sure we're starting them earlier, a little bit earlier than we would expect, maybe, so we can use them as a signal for the first kind of incursions back again. So it's I think there would be spring and fall would be the, and even over the winter would be the times that I'd really focus on ensuring that we have a good surveillance program up and running in wildlife. Yeah.
Thank you. Any publications planned for the results? Those who want to read more about it?
Yeah, we're working on it, so stay tuned.
Great. For waterfowl that are found dead and test positive for avian influenza, are you able to say that the birds died of avian influenza versus a different cause?
That's a great question. And so when we do the surveillance, we do the swabbing. And initially we were doing necropsies as well. So we could look and determine why the animal died. And if the lesions were consistent with avian influenza, we would test tissue for avian influenza to say that the virus is actually in those tissues that showed lesions or evidence that there's something wrong. And so there certainly are some waterfowl or individuals that are dying from influenza, avian influenza, the high pass strain. But we also can use dead birds that die of other reasons as part of our surveillance component, right? So to look for, to look for the virus in animals that aren't necessarily dying. And that's really where the live bird surveillance comes in as well, right? So we do surveillance in those animals that aren't sick. And so we've seen it depends a little bit. There's lots of different species. It depends a little bit on the species. And I think things are kind of changing as they, develop in some cases, some immunity to the virus. So there's a lot going on there, but certainly unlike what you might have expected before this H5N1 situation, there are waterfowl that do die of this, from this virus. And we've seen it, particularly Canada geese, for example.
All right, thank you. For the sampling method that was described, you described the torpedo method and compared it to swabs of birds. Was there any consideration for using other samples for comparison, such as sediment or still water samples?
Yeah, so basically the idea was that we wanted to compare it to the most effective method, which really we get the highest quality with direct animal samples. You're going to get the best virus, you're going to get the most sequenceable virus. And so because we had the opportunity through the banding efforts to obtain those samples, we opted to do so and do that comparison. And now that we know that it's generally comparable, obviously we didn't get as many high quality sequences out of it. But now that we know that's generally comparable, it does open the door for us to do other comparisons as well. And that's certainly something that we could consider moving forward.
Thanks. One other sampling question. For the bird samples, I think you said that they were oral swabs. Were there consideration using fecal samples? There's a lot of virus found there.
Yeah, we do. It's actually like we do an oral swab and also a cloacal swab. So we do get at that end as well. But, and yes, there is interest in looking at fecal samples. I think there's some limitations in terms of, all the, knowing which species and having all the information that you want about that helps us to better really understand the ecology by having a bird in hand, getting all the information about the bird and the samples from that individual that we kind of miss with fecal sampling. But it is something I think that's considered, that people are considering. And I think there maybe are some of the limitations that, associated with all these type of sampling, like in terms of the quality of the sample. But I don't know, Samira, if you or Juliet, if you guys know more about that or have anything to add.
I think it's just a question about the extraction methods as well.
Yeah, that's right. Yeah.
We're just so used to using swab. It's not impossible. We certainly probably could figure out how to process, but then we would have to validate that, whereas swabs, I think, is something that we're generally quite comfortable with. And I guess the other question is, I guess it would be more of an aggregate sample. So that would be the advantage. But otherwise, I don't know in terms of actually picking up virus, if there would be any advantage over a cloacal swab, which would be much more fresh and recent. Yeah. You just don't know if the sensitivity would be affected by something that was sitting in the environment.
All right, thank you. Moving on to the next question. There are areas in the US that are detecting H5 in wastewater, and some suggestion that this could either be from discarded milk or from environmental sources washed into storm drains and then to sewer systems. Does it seem feasible that environmental contamination would be large enough to be detected in this way? And does the torpedo sampling method give any indication of the concentration of virus in the water environment?
Maybe just to address the initial part of the question, I guess it is potentially feasible if they are detecting H5 in wastewater. It really would depend on how much dumping is happening and where they're sampling relative to where the potential water contamination or dumping is happening. In terms of concentration, it was a little bit difficult because the way it worked is that the torpedo was dropped off the back of an airboat. So if you really want to do a precise concentration calculation, you would have to know exactly how many liters of water were sampled. And because it was convenience sampling, I don't know that we really have enough data to know what, I guess I would call it the limit of detection, for lack of a better term, would be. I don't know, Juliette or Claire, if you want to add. add to that, but it was really mainly a pilot project to see if we could pull anything out of the water and whether or not it reflected what was seen in the birds. But concentration and quantitation, I think that will need a lot more work.
And if I think if for the dumping of milk, it's actually, I think the states or various, I think it's responsibility of the state governments, but they do, I think, recommend heat treating the just like before anything is disposed of in the environment. I think there's different regulations for different states, but I think overall there's a recommendation that, raw milk should not be raw milk without treatment should not be discarded. So there is some recognition that could be, I guess, a cause of concern, and there are some guidelines, I think, at least I've seen them, in the US for some of the states.
Thank you. Any thoughts about the viability of HPAI at the sample sites and downstream, and any potential risks to human health?
Yeah, we have not tried to isolate virus at this stage. We just very recently got permission to work with the virus, and if we were to take anything from COVID and wastewater sampling, it's not, viable virus is not routinely isolated from those sample types. So I would anticipate it's a low yield. Is it worth trying at some point? Potentially, but not only is it a risk group 3 agent, but it's also a security sensitive biological agent and still considered a foreign animal disease. So looking at viability would have to be very carefully considered before attempting it. I don't know if others have, but I've not heard that it's been isolated from water, but I could stand to be corrected.
Everything else. So moving on to one more question. How does avian influenza present in waterfowl, and does it change from older versus younger birds?
Yeah, so some birds don't present with any clinical signs, so they can be healthy. Other birds, the most common thing that we see is neurological signs. So that's what we've seen, particularly, like say with the Canada geese and some of the other species that are really impacted. So that there's evidence of neurological signs. So they act strangely. So and they are often, for us, they're found dead. So it seems to be fairly rapid. And if we do catch them before they die, it seems to be neurological signs.
And correct me if I'm wrong, Dr. Jardine, but that's for highly pathogenic avian influenza. Yeah. And so for the lowly pathogenic avian influenza, they tend to not
have any symptoms. We wouldn't expect to see anything.
Or signs.
Yeah, no
signs. Yes.
Yeah. And even some birds infected with highly pathogenic avian influenza show no signs. Yeah.
All right, thank you. I think that's all the time we have for questions today. So as we wrap up today's PHO rounds, I'd like to thank Dr. Samira Mubareka, Dr. Claire Jardin, and Juliette Le-Savoy for presenting. I would also like to thank everyone who joined us for today's webinar. To access past PHO presentations and to view confirmed and upcoming sessions, please visit the PHO website, head to Education Events, and click on Presentation. Thank you, and have a wonderful day.